Hi everyone, today we're going to do something just a little bit different. I'm going to talk about a series of studies that were performed by Hikosaka, Miyashita, Miyachi, Sakai, Liu, Rand, and Karate in the mid to late 90s. So the overall goal of these studies was to look at the learning of procedural sequences. Now, procedural sequences are interesting because they're something that we use every day, all the time. A procedural sequence could be something like playing a song on the piano, which is a sequence of many individual motions of the hand strung together in a sequence in order to create a finished action. And you can imagine that this type of procedural sequence is something that is used all over the place in your everyday action. Something more mundane might be like driving to work, which is composed of a series of procedural actions: turn here, turn there, go straight at this place, go across the bridge. And this series of actions put together creates the sequence that allows you to get to work in a really efficient and procedural way. So let's take a look at how these researchers chose to address this issue. So they trained monkeys to perform what they called a 2x5 task. In this task, the monkey is sitting in front of a 4x4 array of buttons, and at any given time, the monkey will have two buttons that are lit up, and the monkey has to press these two buttons in a particular order. So you can see in the figure here that if these two buttons are lit up, then the monkey would have to press the bottom one and then the second one, button 1 and then button 2. If the button gets these two correct, then the monkey will move on to the next set of buttons. These two have to press those in order as well, and then move on to the next set of 2 and the next set of 2 and the next set of 2. And they say that this set of 5 sets composes a hyperset. So that's going to be the terminology that's important to think about looking at these studies moving forward. If at any time the monkey gets an individual set wrong, so presses the buttons in an incorrect order or presses a button that is not lit up, then the monkey has to press the home key in order to start the whole hyperset over. And the monkey repeats the same hyperset over and over again until they amass 20 correct trials on that hyperset, at which point they move on to the next hyperset. And in order to keep the monkey motivated, they're being given a small amount of juice every time they get a set correct. And that juice is just being delivered in like a little straw, basically, directly to their mouth. So let's take a look at some of the data. So here we can see the learning by a monkey PI over the course of 30 days on a particular hyperset. So that's one thing I should have mentioned. They have the monkeys learn the same hyperset repeatedly for many days. And that's not to say this is the only thing this monkey is learning. One of the big advantages of this task is that the monkey can learn a near infinite array of different hypersets. You can imagine that there's really a high ceiling on the number of different 10-button sequences that can be composed in this 2x5 task. And you can see here that when this monkey was initially facing this particular hyperset, it took the monkey about 80 trials to amass those 20 correct responses. And in fact, as I'm looking at this in a little more detail, you can see that there aren't even 20 full sets of 5 in 80 trials. So the monkey's really having a tough time here, and they're getting a lot of trials wrong. By day 2, you can already see that performance has increased quite markedly to where the monkey's now reaching that criterion of 20 trials correct over the course of their experience with this hyperset in under 50 trials. And then by day 3, you can see the monkey's getting even better. They're reaching that criterion to move on to the next hyperset in only 35 trials. And then if we fast forward out to day 30 here, you can see that the monkey is only getting 2 trials incorrect. And in fact, on the very first trial that they see this particular hyperset, they're already getting it correct. So they really do demonstrate mastery over these hypersets. You could imagine day 1 is kind of the first time you try and play a song that you've never seen before, and day 30 is after you've been practicing that song over and over and over again. And so they wanted to take a look in more detail at how this learning was going on. So they measured learning in 2 key ways. One is going to be the number of trials performed relative to the number of successful trials, and specifically here we're going to look for the number of trials performed that are required in order to get 10 successful trials. So for example, you can see that on day 1, shown with the filled in circles out here, it's taking the monkey somewhere in the region of 65 to 70 trials in order to get 10 of them correct. The monkey's really not doing super well. But by the time we're out at day 30, this line all the way out to the left, you can see that the monkey is getting up to 10 correct trials in only 12 trials, implying that they only get 2 wrong. And you can see those 2 wrong indicated here by the little horizontal motion of this line. The other way that they're going to measure is going to be in the speed that, or the amount of time that it takes the monkey to amass those 10 correct trials. And importantly here, they're only looking at the correct trials. So even though it might take the monkey 65 trials to get a total of 10 trials, they're only going to count the time that it took the monkey to get those 10 trials. And that's why these lines end up being quite close together, even though we're looking at really different levels of performance. But one thing that's really interesting to think about here, so 10 successful trials, that means 100 button presses. And what we're seeing on day 30 is that the monkey's getting 100 button presses correct in about 30 seconds. So that's implying that this monkey's doing 3 button presses per second. So you really have to imagine the monkey's sitting there in front of this keyboard, this 4x4 keyboard, and they're just like, you know, crazy fast, just total expert. They know exactly what buttons to press. They've really memorized these sequences. So just to kind of conceptualize what these two measures are telling us, this trials to criterion, trials to get number of trials to get 10 successful trials. That's going to kind of be a measure of knowledge. So trials to criterion, a measure of knowledge. And then down here, the speed is going to kind of be a measure of fluidity. And anyone who plays a musical instrument, I think, can appreciate the difference between these two things. Knowing what the next chord or note to play is, and being able to fluidly move to that next chord are kind of two different things. So how does this learning progress over the course of time? Now that we have these metrics, we can take a look at this in a little bit more detail. So here we're mapping the acquisition of a single hyperset. Again, this is monkey PI that they're showing us here. And you can see, as we've kind of already seen, the number of trials to criterion, it really precipitously drops, even from day 1 to day 2. There's this very quick drop right at the beginning, and then it looks kind of flat from there on out, especially from day 10 onwards. It looks pretty flat. But what they've done in this right panel over here is they're showing us the exact same data, this trials to criterion measure, but they're showing it to us in a log scale. And what this does is it kind of blows up what's going on out here in the later stages of learning. And what you can see is that, yeah, there is this really steep learning at first, but there's actually continuous learning going on at a slower rate for many, many days. So the monkey is just very slowly but continuously zeroing in on getting right down to the 10 trials in a row correct right off the bat. And this dotted line, that's indicating the number of that 10 trials is kind of the minimum for this measure. Since the trials to criterion, 10 successful trials, the minimum number of trials it's going to take you to get 10 successful trials is 10. And so that was just one. Sorry, my cat. She's kind of being a little bit of a pest. That was one particular hyperset. This figure just shows you that this does indeed happen with other hypersets. You can see that precipitous learning initially, followed by kind of continuous slow learning over the course of days. One thing to notice, look at this x-axis. They tested these monkeys for about a year on this task, and that really lets you know just how much we're really looking at expert level behavior from these monkeys. Now, one thing that was super interesting, we've just looked a lot at how learning of individual hypersets went. Now we're going to look at the learning of new sequences. So when they present a monkey with a hyperset that they've never seen before, how's the monkey going to do? And what we can see is that early in the monkey's experience with the 2x5 task, they're kind of all over the place. Sometimes they're taking way out into like 150 trials to reach that criterion of 10 successful trials. But by the time they get out to having done this for coming up on a year, they're getting pretty tight in their performance to where it's always taking them somewhere in the region of 20 to 60 trials, and they're really getting very consistent. So not only did we see that learning going on for individual hypersets, but there's actually facilitation going on for novel learning of new hypersets as well. And this is kind of like, once you know how to play a musical instrument, you can learn new songs on it easier than you could learn new songs when you were first starting out. And then the final thing that's kind of interesting to note in these data is that old sets are stored even without practice. So here we're looking at, on the top panel, performance of hypersets after they have not been practiced for an entire month. And on the bottom panel, we're looking at hypersets that were learned in the very same behavioral testing sessions that had never been seen before. And what you can see immediately is that these relearned ones in the top panel shifted over to the left, indicating that they're making fewer errors. The number of successful trials as a function of the total number of trials performed is much higher. So overall, this study demonstrates there are 3 types of learning going on in this procedural 2x5 task. First, there's fast learning of individual sequences that happens within 1 or at the most a few days, that kind of precipitous fall in the number of trials to criterion. Second, there's slow learning of individual sequences over many days. So after that initial precipitous fall in the number of trials to criterion, we saw that there was a slow but steady approach to getting all the way down to perfect behavior. And that took a long time. So there's this kind of initial fast drop, and then there's slow, continuous learning that keeps on amassing over many days. And that information is stored also over the course of periods that may not even include practice with those particular sequences. And then finally, and really interesting, there's actually slow learning of the general task. So something generalizable about this skill of learning sequences is somehow being accrued by the monkey. So the next thing in the next couple papers I'm going to describe, this group of researchers, Hikosaka, Rand, Miyachi, Miyashita, and a few others, wanted to know how can this learning be supported by the brain. So the first area that they looked in the striatum, an area of the basal ganglia that's known to be important for motor behaviors, habits, rule-based activity. This is an area that's affected by diseases like Huntington's, ALS, Parkinson's, really important area of the brain. And what they're going to do is they're going to put injection needles into areas of the basal ganglia, and they're going to inject mucimol, which is something that will, for a short period of time, inactivate that part of the brain, kind of turn it off, and create a short-term lesion to this area. Doesn't cause any long-term damage, but allows us to take a look at what is the contribution of this area by looking at how the monkey's performance may get worse when this area is no longer functioning. So they had a few different areas of the basal ganglia that they're going to inject. Here we can see the caudate, this kind of middle bit, and the putamen, this kind of lateral bit, in 3 different drawings of a monkey brain. We're looking at it coronally. And you can see again the caudate, the putamen, the caudate, the putamen. And they had 3 different groups of injections. So they would either inject into all of these cross-indicated locations, and that would really inactivate the entire anterior part of the striatum. The striatum is this combination of the caudate and the putamen. That would inactivate this whole anterior part. Then the other area that they would inject the open circles. You can see that in both of these. So that's injecting into the middle and posterior caudate. And then they would also inject into the closed circles, and that's going to be the middle and posterior putamen. And they're going to do these 3 different sets of injections at different times so that they can look at the different effects that these injections may have on performance. So first, they looked at how new hyperset learning was affected by these injections, and there's kind of a standout effect here where injection into the anterior striatum is going to cause a significant increase in the number of errors that both monkey PI and monkey ME are gonna produce when trying to learn new hypersets. And this is really pretty dramatic. When you look at the control, which is an injection of just saline, you can see that the monkeys are taking somewhere on the order of about 15 or 10 trials or they're making about 15 or 10 errors on their way to reaching criterion. And that number just about doubles for both of these monkeys when they have this inactivation of the anterior striatum. So this area is definitely playing a role in new learning. Then, by contrast, we can take a look at the effects on learned hypersets. And here, notice that the y-axis is a lot smaller. There's way smaller number of errors going on in the learned hypersets. These are the ones the monkeys are experts on. They've done them, for 30 plus days. But we can see now the putamen, so that's these filled in circles, when that area is injected, suddenly there's an increase in errors on these learned hypersets, and that's a different effect than what we saw for the new hypersets for the putamen. So inject the putamen and new hypersets pretty much unaffected, but learned hypersets affected where we have an increase in errors. By contrast, inject the anterior striatum, both the caudate and the putamen, and you can see an increase in errors for new hypersets, and you do see an increase in errors for the learned hypersets as well, but the effect is a little bit more modest. So overall, going back to our three types of learning, that fast learning of individual sequences or new sequences, we can see that the anterior striatum is important, and then the slow learning of individual sequences, or maybe we should say the remembering of previously learned sequences, is supported by the middle or posterior putamen. One thing to caution, though, these effects are kind of small. It's not like the monkey couldn't do one or the other type of learning with these injections. They could still perform the task, but it just took them a little bit longer. They made a few more errors. So the next area that they wanted to take a look at in a separate study was this pre-supplementary motor area. And here we're looking at a lateral view of a monkey brain. You can imagine an eyeball right here. The monkey's looking forward. And up here, we've got the supplementary motor area. It's kind of like tucked in, kind of over the midline of the brain, right in front of the primary motor cortex. And then there's this pre-SMA or pre-supplementary motor area that's just a little bit in front of that SMA region. And they're going to go ahead and take a look at how does this pre-SMA versus SMA, how do these regions contribute to the learning of this task? So they did the same mucimol injection method. Now, I'm going to caution you, this was only tested in one animal. So unlike in the stuff we've looked at so far, where 2 animals were tested, so you can have a little bit of replication going on, this is just one animal, so keep that grain of salt in mind. But we can see a pretty dramatic effect. So for learned hypersets, these are the ones the monkey's an expert on, no effect of these injections, either the pre-SMA or the SMA, doesn't matter. But then for new hypersets, we can see that the pre-SMA injections of mucimol there have a really pretty dramatic effect where there's a much larger number of errors when injections are done into the pre-SMA than into the SMA. There is a modest effect of SMA injections, the supplementary motor area, but it's definitely not as big as the injection into the pre-supplementary motor area. One thing kind of interesting to note, this particular monkey was kind of an expert because you can see he's learning new hypersets in with only about 8 errors, which is smaller than what we saw for the 2 monkeys that were shown in the previous graph. So that's just a note on how individual variability between monkeys is definitely a real thing, and so it is tough to interpret these data from only one animal, but still this effect is pretty strong with the difference between new hypersets and learned hypersets. So then they also took a look at what neuron firing looked like in the pre-SMA, and you can see, corroborating the effects of the mucimol injection, that this area of the brain, at least for this individual neuron, now this is the only neuron I could find in any of their papers that they actually published from these recordings. I'm assuming they recorded more than one, but again, this is definitely not data that they worked up to the point of having a full paper for it. This was actually something that they published only in kind of a small review. But what you can see is a pretty dramatic difference for this particular neuron between new hypersets and learned hypersets. So this neuron, as the monkey moves from set 1 to set 2 to set 3 on to set 5, in this particular hyperset, this neuron's just going crazy, firing all the time. What we're looking at here in these rasters are just the little black marks indicate the spike of the neuron, and each row in this little figure down here indicates one particular run through the hyperset, with the top histogram representing kind of the averaged activity across all of the trials. And then down in the bottom panel, we're looking at learned hypersets, and you can see the neuron really just doesn't care. So this kind of corroborates the idea that was seen with the mucimol injection, that the pre-SMA is particularly important for new learning, not so much for learned hypersets. So going back to our 3 types of learning, it now looks like this fast learning of individual sequences is something that is controlled by the anterior striatum and the pre-SMA, but not the SMA, since the effects of mucimol injection there were much more modest. And in fact, this kind of makes sense because the anterior striatum is known to have anatomical connectivity to the pre-SMA more so than to the SMA. And again, from that previous study, we saw that the middle posterior putamen was important for the learned sequences that the monkey's an expert on. But still no word on that slow learning of the general task. So next, they turned to the cerebellum in another study. And here in this figure, we're actually looking at a photo of the monkey's brain that was taken post-mortem, looking at the striatum, and we can, or sorry, not at the striatum, at the cerebellum, and we can see these kind of vertical tracks right here indicated by the mouse arrowheads in the picture. These are actually the tracks that were that indicate where the injection cannulae for the mucimol injections actually went. So when they were done with doing the mucimol injections, they passed a small amount of electrical current through an electrode at the same location that they had done the mucimol injection in order to make a small lesion that would be visible when they took the monkey's brain out after sacrifice in order to localize where they had done the mucimol injections. So this is really showing us just gold standard proof that they did in fact inject the mucimol exactly where they said they did. So if you look here, you can kind of see there's this kind of backwards C-shaped area that's a little bit darker gray than the area around it, and then in front of it there's another little gray area, and then another little gray area further medial to that. So these areas are going to be where they're going to really be focusing on, and in particular they're going to focus on this backward C-shaped area called the dentate nucleus of the cerebellum. Aziza, you're getting in the way. Okay. And over here in this drawing, they're showing us where they did all of their mucimol injections. So we're going to get a pretty good sense of how these different areas of the brain contribute to this task. And what we can see now is, unlike with the SMA, the pre-supplementary motor area, the pre-SMA, you can see that it's the learned hypersets that show an effect when injections are made into the cerebellum. And in particular, these black filled-in dots are showing us which injection locations showed this significant effect. So here we're looking at the number of errors that are made when the monkey is performing learned hypersets, and you can see that it's increased when mucimol injection is given, as indicated by the hashed bars as opposed to the open bars. And this is a specific effect only to the ipsilateral hand. And this is just because the cerebellum controls the right hand with the right cerebellum and the left hand with the left cerebellum. This was actually news to me. I did not realize that the cerebellum is not crossed in the way that the eyes and the whole cortex is in motor control, and in somatosensation. So that's interesting. And then we can see for new hypersets, there's just no effect. So this is really pretty robust, because unlike what we saw for the SMA data and for the striatum data, where all of the effects were kind of, yeah, they do have a preference, but they also kind of affect the other one as well, here we see that it's really only the learned hypersets. It's a very specific effect. I think we can be pretty confident looking at these data that the dorsal aspect of the dentate nucleus of the cerebellum is involved in the learned hypersets, but not in the new hypersets. And this is replicated across 2 different monkeys. So all the things that you want. So 3 types of learning overall. We found that we saw in these series of studies that the fast learning of individual sequences, that is that learning that happens within 1 or maybe at most a few days, it's dependent, or at least the anterior striatum and the pre-SMA are involved in it. I shouldn't say dependent. but the supplementary motor area itself does not seem to be so involved. The slow learning of individual sequences over many days, that seems to be something that's more mediated by the middle and posterior putamen. And then probably the strongest data that we really saw was the dorsal area of the dentate nucleus of the cerebellum. That seems to be really playing a role. And that's interesting because the cerebellum is also involved in things like walking, stuff that you just don't even think about. So the fact that the cerebellum is playing this role in these learned sequences indicates that for the monkey, by the time they're doing these well-learned sequences, they're not even thinking about it. This is just totally automatic. You can imagine driving to work when it's a route that you've done many times. Sometimes you just arrive at work and you don't even know how you got there. It's that kind of thing. Or playing a song on a musical instrument that you've played many, many times where you don't even have to think. Your hands are just moving to the next set of notes. And in fact, in some separate data that I didn't present to you, they showed that the monkey's eyes for these well-learned sequences actually move to the next button that they're going to press before the button even lights up. So the monkey has completely memorized this set of 10 button presses. It's no longer 5 sets of 2 little mini sequences. It's actually just a series of 10 button presses all together. So that's super cool. One hole in all of this, though, where does that slow learning of the general task over many days happen? Still unknown. And in some ways, that's kind of the coolest thing, because what we really want to know is how do you learn one very specific thing, one specific set of 10 button presses, and then make it into a general skill? How is it that you can practice several songs on an instrument and all of a sudden you're just better at playing other songs that you've never seen before? So really a beautiful series of studies from Hikosaka, Miyachi, Miyashita, Liu, Rand, and I know I'm forgetting a name or two. I apologize, but still I am left wondering where does that slow learning of the general task happen. So hopefully we can find something to take a look at in future weeks. And thanks for watching. Okay, bye.